So we're picking up where we left off. Now we're starting with cellular respiration. So the battle against world hunger can use plants adapted to water scarcity. Evolutionary adaptations in some plants, we see how they can thrive in very hot, very dry conditions. These adaptations allow for less water evaporation coming out of the holes underneath the leaf, and those holes are going to be referred to as the stomata. And so first, to understand that, we have to understand how plants use water. Plants are going to take in water from the root, and of course it travels up through the stem and to the leaves. The leaves are then going to allow the evaporation through the stomata that's going to be in the leaf. So that's going to be a small hole that's found within that leaf. So that stomata, when it's closed, does not allow that water to come out. If it's open, it can't come out. So some plants have adapted to where dry, hot days, that stomata closes and it doesn't lose water. So the ideal leaf is going to be a leaf with a large surface area that's able to capture a lot of sunlight. And it's also going to be very porous. This is going to be because it needs carbon dioxide to come in order to perform photosynthesis. With that carbon dioxide and the water it's pulling in from the roots and then the sun, it's able to convert all of that into glucose. And then it's going to release its byproducts as oxygen out of those pores. So here's a look at that stomata. These are the actual pores that are going to be used for gas exchange. So how to get that CO2 in when the stomata's shut? That's going to be the big problem. So it might be a nice sunny hot day. That sun should be great for creating lots of food, lots of glucose through photosynthesis. However, if it's hot and dry, it's going to need to close those stomatas in order to keep from losing too much water, because that will then kill the plant. So what it's going to do is it's going to be able to close the stomata, but then it's got to come up with another way to get that carbon dioxide to help create that glucose. So when the stomata closes, the CO2 levels, like we said, are going to drop, and the oxygen levels inside are going to rise, because not only can it not get carbon dioxide in, it can't let oxygen out. So what it's going to use is a carbon-fixing enzyme that's going to combine with the oxygen instead of carbon dioxide with what is called a RUBP, and that's going to be called photorespiration. So hot, dry weather causes that stomata to stay closed. Oxygen levels, like we said, are going to rise. Carbon dioxide levels fall. That photorespiration is going to be very common under these conditions. If it lasts too long, however, plants can die from lack of glucose synthesis. Remember, it does store extra glucose as starch in the plant, and it's going to go to that starch and use that starch just like we use adipose tissue. So here we see it's referred to as C4 photosynthesis. You have your carbon dioxide. During the day, the stomata are open or slightly open, and it's going to go through a carbon uptake cycle. It's going to be able to create ATP. It's going to go through that Calvin cycle and perform the ability to make your sugar or glucose molecule. The advantage to this, water loss is going to be minimized in those warm climates. But the disadvantage, it's actually going to take more ATP in order to produce the sugar molecule in the end. So the C4 plants are going to produce what's referred to as a CO2 sticky tape enzyme, and it's going to add an extra set of steps when it does this. So because it adds that extra set of steps, it requires more energy. And so it's going to use more energy than the C3 pathway, which we'll talk about in a minute. So C4 plants thrive when light's abundant, but water is scarce. So you'll see these in places like deserts and hot climates. You can find it in things like in the grasslands. So you'll find it with corn. You can also find it with sugarcane, sour gum, crabgrass, and some thistles. So we have 3 photosynthetic pathways. We just talked about the C4. You see how it adds that extra cycle in with the carbon uptake. The C3 photosynthesis is going to be very efficient. Water lost is going to be lost through evaporation. And so this is going to be the type of photosynthesis we have in our area with a temperate climate that can be hot, it can be dry, but it also can be cold. And it receives varying amounts of sunlight just depending on what season we're in. Then you'll go into what's referred to as CAM synthesis. CAM synthesis is going to be the ultimate photosynthesis for very, very dry areas such as deserts. And so it's also going to use this second step of steps that it has to go through, and it does require more energy. So advantages, water loss is minimized, and that's what we're going for. Disadvantages, again, it requires more energy, and this also, because it requires more energy, is going to cause very slow growth in these plants. So they won't grow near as fast as the C3 and the C4 plants. So environmental conditions with those C3 plants, like I said, remember they're going to thrive where water is abundant, or if light levels are low. So we can have cool, wet, and even cloudy climates where these plants will still do fine. So these are going to be most of the trees like we see, the deciduous trees and the conifers. It's going to be things like wheat, oat, rice, and Kentucky bluegrass. Here's a distribution of photosynthesis methods around the world. Going into that CAM photosynthesis, it's going to be what you see like with the same steps that we went through with the C4 photosynthesis, except it's going to be able to open up its pores and take in some of that carbon dioxide at night, but it's still going to require a lot more energy to do this. So it's going to close its stomata during the day, opens the stomata at night. It's going to allow that CO2 to get in, and it's going to be temporarily bound by a holding molecule, which is going to hold it and slowly use it throughout the day. And then it's going to release its oxygen when it opens up at night. So with this, the C4 and CAM photosynthesis are going to be evolutionary adaptations at a biochemical level that allow plants to be able to grow when it's very dry and very hot outside. However, it is more energetically expensive than the regular type of photosynthesis that we see in our area. Cellular respiration is going to convert food molecules into ATP, which is going to be our universal source of energy for living organisms. So we see here she's holding a nice, looks like a huge hamburger, and that's going to be full of energy molecules that her body's going to be able to convert, and then it's going to change it over into a form she can use, which is going to be ATP. Right now, it's going to be a stored energy, which is going to be found as a chemical, and that's what we refer to as our calories. How do living organisms fuel their actions? And well, they're going to use cellular respiration in order to do that. So remember ATP molecules. That's going to be what we use in order to get things done. So that is our cellular currency. That's our energy, or like our little battery for our bodies. The currency exchange this time is going to be food molecules that are going to be converted over into ATP, and we're going to use a process called cellular respiration to do this. So we converted sunlight into food or stored energy through photosynthesis. Now we're going to take in that stored energy as food and convert it over into ATP, which is going to be our cellular exchange or cellular energy. So efficiency is going to be important, but unfortunately the first step is very inefficient. It's referred to as glycolysis. So this is what's going to start off cellular respiration. In order to even start off, it has to use 2 ATP to even start to process the food you took in to get energy or to make more ATP. So this is going to occur in the cytoplasm, somewhere near the mitochondria. And so it's going to take a glucose molecule. It's going to break that glucose molecule in half and create 2 pyruvates. When it does this, it actually creates 4 ATP. But only 2 ATP are actually going to be able to be used, because then 2 ATP have to return back to the glycolysis stage and start it all over again. So out of 4 that it's going to release, you only get to use 2. Then we're going to have what's referred to as the Krebs Cycle, followed by the electron transport chain. Those are going to occur in the mitochondria. And so it's going to take and make Acetyl Coa Production. And that's going to be a preparatory step that it has for the Krebs Cycle. So it's a 3-step process. In the end, it's efficient. It gives you 90% of your ATP, though, at the end with the electron transport chain. So starting off, number 1, we have glycolysis. We took the glucose molecule from the food. We split it in half, created 2 pyruvates. We got 2 ATPs there. Then it's going to go into what's referred to acetyl CoA production. So it takes that pyruvate. It's got that acetyl carbon dioxide that's going to release, create that acetyl CoA. It's going to come on down to the Krebs cycle. It goes through the cycle. It releases more carbon dioxide. It creates some ATP there, but it also creates 2 high-energy electron carrier molecules. It's going to create NADH and FADH2. Those are going to go into the electron transport chain, where it's going to help carry those electrons through and help create the most abundant amount of energy from the food that you just ate. So this is where 90% of it's going to come from. Also in this process, we take in the oxygen with it, and we're going to release water and some of those ATPs. So cellular respiration requires fuel, and it also has to have oxygen in order to do this. So that oxygen is going to be needed by the mitochondria in order to create your ATP. So potential energy, like we said, stored in chemical bonds of sugar, protein, and fat molecules, which we get from our food. We break those bonds to release the high energy electrons that were captured in the ATP. And we have the oxygen there that's going to be the electron magnet. So it's going to capture those and help put them into bonds. Input for cellular respiration, oxygen and sugar. Output, or what we get after we go through the cellular respiration process, is going to be carbon dioxide, water, and energy. So with a human example, you eat food, let's say you eat dinner, you're going to digest it. From there, once it exits the stomach, it goes into the small intestines where it's going to start to be absorbed into the bloodstream. That bloodstream is going to deliver those nutrients or those different molecules that you might need to the cells. Those cells then, once they take it in, start the process of breaking it down. So in the cytoplasm, as it's making its way to the mitochondria, it'll go through glycolysis. Then once it enters the mitochondria, it will go through the Krebs cycle and then the electron transport chain. In the end, you have your ATPs. So just looking at that again, that was that process that we went through. So in summary, living organisms extract energy through a process called cellular respiration, in which the high energy bonds of sugar and other energy-rich molecules are going to be broken. That releases the energy that went into creating them, that energy that was stored from the sun. So the cell is going to capture the food molecules, stored energy, in bonds, like we said, of ATP molecules. That requires fuel in order to even make your ATP molecules or your energy, along with water and carbon dioxide at the end. So the first step of that cellular respiration, like we said, was glycolysis. It's going to be the universal energy releasing pathway. So anything from plants, like you see in the first picture, to animals in the middle, and even bacteria on the right are going to use cellular respiration in order to get their energy. Now you might think plants, for example, get their energy from the sun, but they don't. They're actually getting the energy to make food through the chloroplast, and then they take that food that they made, and then it has to go through cellular respiration in order to use the energy. So that's why glycolysis is going to be considered the universal energy releasing pathway. So with glycolysis, we have 3 of 10 steps that are going to yield energy. It quickly is going to be harnessed to make that ATP. We're going to use things like high energy electrons that are going to be transferred to NADH, which is your high energy electron carrier. As a result, I told you get 4 ATP. You broke that glucose, and now you've got 2 pyruvates that you made, and you have 2 NADH molecules that are going to store high energy electrons. So with those 4 ATP, remember 2 have to go back to the next glycolysis that's going to get ready and start again. So you only technically get 2 usable ATP. So looking at it, here's just a visual of what we just said. It takes 2 to actually start. So it takes 2 ATP, which you see right here, that actually start breaking this glucose. So that actually starts with the glycolysis here, breaks the glucose, it takes in the 2 ATP, creates 2 pyruvates here. So you got one, and then you have the other one. Once you break those, it's going to be able to release 4 ATP, 2 NADHs, and some water. And in the end, you also have 2 pyruvates. So what you got as a product, 4 ATP, 2 NADHs. But remember, you can't use all 4 of these ATP. You're only going to get to use 2, because 2 of those go back to the next glycolysis that's getting ready to start. So glycolysis is the initial phase in the process by which all living organisms are going to harness energy from food molecules. Glycolysis occurs in a cell's cytoplasm and is going to use the energy released from breaking chemical bonds in food molecules to produce high-energy molecules, ATP and NADH. The second step in cellular respiration is going to be what's referred to as the Krebs Cycle. This is going to extract the energy from the sugar. So we had the glucose, we broke it into 2 pyruvates, and so each pyruvate molecule is going to pass high energy electrons to NAD+, creating NADH. Then a carbon and 2 oxygen atoms are going to be released as carbon dioxide. So this is going to be this is what you're going to use or have to breathe out as a waste product. Then coenzyme A is going to attach itself to the remaining molecule, creating what's referred to as Acetyl Coa. So now we have that Acetyl Coa that we just made ready to move into the Krebs cycle. So here we're going to have a new molecule formed. Basically what's happening, the Acetyl Coa molecule from the glycolysis is going to enter the cycle and bind to the oxaloacetate, and it creates a 6-carbon molecule. And so you see, hold on just a minute, you're going to see that 6-carbon molecule here. From there, high energy electron carriers, NADH, are going to be made, and carbon dioxide is going to be exhaled. So we took the NAD+, you're going to add a hydrogen, creates NADH, and you're going to exhale some carbon dioxide there. And it's actually going to be 2 carbon dioxides that you see that are going to be ready to be exhaled. So from there, we're going to move on. You got that oxaloacetate that is going to be reformed. ATP is going to be generated, and more high energy electron carriers are going to be formed here. So the remaining 4-carbon molecule is going to be rearranged to form the oxaloacetate, and in the process ATP is formed. Electrons are then going to be passed to the NADH and the FADH2. So here we had our 4-carbon molecule, we made some energy, we took the NAD+, added a hydrogen, created NADH, took the FAD, added 2 hydrogens there, and made FADH2. It's going to go through this cycle 2 times. So with this process, you're going to create another 2 ATPs. So the payoff from the Krebs cycle, like we said, 2 ATPs, you're going to end up with 8 NADHs and then 2 FADH2s. So in summary, a huge amount of additional energy can be harvested by cells after glycolysis. So far we haven't got a lot of energy. We have 4 usable ATPs. So first, the end product of glycolysis is pyruvate. It's going to be chemically modified. Remember, we're changing it to acetyl CoA. Then it's going to enter the Krebs cycle, where the modified pyruvate is going to be broken down step by step. This breakdown process is going to release carbon molecules to the atmosphere. That's when you exhale and breathe out, as bonds are going to be broken and capture some of the released energy into 2 ATP molecules and numerous high-energy electron carriers. Remember your NADHs and your FADH2s. The third step in cellular respiration is going to be where your ATP is mainly going to be built. This is the one that's going to give you the most bang for the buck, the most payoff. So two key features of the mitochondria are going to be essential for this. Feature one is referred to as a bag within a bag structure, and so that's going to help with this process. And then feature two, the electron carriers are going to be organized within this bag system. It's going to be found in the inner bag, which is going to help harness the energy. So first step, looking at this bag within a bag, we look inside the mitochondria, and it's going to either lay inside the intermembrane space or the mitochondrial matrix. That intermembrane space is between the two bags, and that mitochondrial matrix is inside of the inner bag. Now, in that inner bag, it's going to be studded with molecules. They're going to create electron transport chains that are going to allow for that ATP production. So in order you're going to figure out how to make this ATP, you're going to follow the electrons. And so remember, inside the mitochondria, so you're going to look at that intermitochondrial matrix. It's going to travel down towards that inner membrane space. So high energy electrons are passed from carriers of NADH and FADH2 to a series of molecules embedded within the inner membrane called electron transport chains. At each step in the electron transport chain, the sequence of handoffs is going to be the electrons as they fall to lower energy states. When they do this, this is where they're releasing energy. So every time they start to fall, energy is released. At the end of the chain, lower energy electrons are going to be handed off to the oxygen, which then combines with hydrogen ions that are there. And when you put the oxygen and hydrogen together, you're going to create water. That's also going to be something that you're going to use to exhale. The energy is going to be used to power proton pumps, which pack hydrogen ions from the mitochondrial matrix into the inner membrane system space. And then those protons are going to rush back into the mitochondrial matrix with greatest kinetic energy, and there it can be used to build the ATPs. Proton gradients and proton potentials. So the force of the flow of hydrogens, they're going to fuel that attachment of free-floating phosphates that we're going to find here. And so you're going to start off with a molecule called ADP, adenine diphosphate. And because of these hydrogen ions, they're going to be able to attach a third phosphate on and change it over to adenine triphosphate and make it ATP. So the largest energy payoff of cellular respiration comes as electrons go from the NADH and FADH2 that are produced during glycolysis in the Krebs cycle as they move along the transport chain. The electrons are going to pass from one carrier to another, and energy is going to be released, and that's going to pump protons into the inner membrane space. Those protons are going to rush back into the inner mitochondrial space, and when they do this, it's going to act as like a fuel, and it's going to help create these ATPs from ADP. So just a summary, we went through glycolysis. Then we had our acetyl Coa production. We went into the Krebs cycle from there. And then from the Krebs cycle, we go to the electron transport chain. All in all, you get around 32 ATPs. So with this electron transport chain, on average, you get around 28 ATP. Or 26, just depending. It's actually 34 if you think about it, but then we have to put 2 back into that glycolysis. So there's going to be alternative pathways to energy acquisition. Beer, wine, and spirits are going to be products of cellular metabolism, but these are going to occur in the absence of oxygen. So remember, in order to make ATP at the mitochondria, it does require oxygen, but sometimes oxygen's not going to be available. In the case where electrons or oxygen's not available, you're going to see electrons generated from the process of glycolysis in the Krebs cycle. If it's present, we're going to follow into this pathway. Electron acceptors are going to be there with your oxygen. End product, you create water. However, if oxygen is going to be lacking, think of things like exercise when you're running or if you start to feel the burn. What's going to happen is that burn is going to be created inside the muscle because the oxygen's not present. So when it starts to build up, instead of having an end product of water, it's going to create what's known as lactic acid. That acid gets between the muscle fibers, and as they're moving and contracting, it starts to cause a lot of friction, and that friction is what makes you feel the burn. So the end product here is going to be lactic acid. You're able to clean that out of your body through heavy respiration to help break it down and get rid of some of that carbon dioxide, or you can also help break it down in the kidneys. Another way, in some organisms, they lack oxygen, so we have things such as yeast, and they're going to use electron acceptors to make acetaldehyde, and then their end product will end up being ethanol. So think about yeast being used to make bread. It's going to release carbon dioxide and ethanol, and that's going to actually cause that bread to rise. Yeast is going to be used in the production of beer. It's also going to be used in the production of wine because it's going to be able to take the glucose from the foods or the fructose, and it's going to be able to change that over into a usable energy source for this organism, and its end product is going to be that fermentation, and the fermentation is what's creating the alcohol. So in summary, oxygen deficiency limits the breakdown of fuel because electron transport chain requires oxygen as a final acceptor of the electrons generated during glycolysis and the Krebs cycle. When oxygen is unavailable, yeast or organisms like them are going to be able to go through a fermentation process. This is where they're going to use different electron acceptors, pyruvate, and they're going to be able to generate ethanol in the process, which is going to create the alcohol part of beer, wine, and other alcoholic beverages. Eating a complete diet is going to be able to help your cells run properly. And so looking here, energy from fats, carbohydrates, and proteins will go through certain processes in order to help you create your ATP or your energy. So with fats can be broken down into fatty acids. If they're broken into fatty acids, they can go directly into that acetyl CoA production. So they come straight down here and they'll pick up acetyl CoA, then to the Krebs cycle, then to the electron transport chain. If they're broken down into glycerols, the glycerols go through glycolysis. There you create the two pyruvates, then it goes into the acetyl CoA, then the Krebs, and then electron transport chain. If you're going through carbohydrates, first it breaks it into simple sugars, then it goes through the process. If you're eating proteins, it gets broken into carbon compounds. Some of those carbon compounds will hit that glycolysis here and then go through the process, and some are going to be turned into an amino group. The amino group is going to be used in the production of tissues, or it can be excreted as waste, like urea. So in humans and other organisms, they're going to have metabolic machinery that allows them to extract energy and other valuable chemicals from all of our macromolecule food sources, like our proteins, fats, and carbohydrates. And that's going to finish us up for this unit. We'll see you next time, starting in on DNA.